Good morning, everyone. Today we have Professor Post from Netherlands giving a great talk. He is a MD, Ph.D. and a professor of physiology at Maastricht University in Netherlands. His main research interest is in the engineering of tissues for medical applications and for food, which has led to the development of cultured beef from bovine skeletal muscle stem cells in an effort to supplement and transform the traditional meat production through livestock. In August 2013, he presented the world's first hamburger from cultured beef. He is CSO and co-founder of two companies, Mosa Meat and Corium, that will commercialize cultured meat and cultured leather. The floor is yours, Professor.
Thank you, Mehmet, and wonderful to be here. I admire you that you're all up this early to follow this lecture, and I'm extremely happy to share some of the data with us. As indicated, I'm also Chief Scientific Officer of Mosa Meat, so as just a disclosure, I have a commercial interest in this as well, and it also means that some of the data may be presented in a little bit more hidden way than you would expect normally from a purely scientific exposure. So just as an overview and to just steer your minds towards what does it mean to apply tissue engineering for food and how does it differ from the medical applications. And there are a couple of very obvious differences, and they basically dictate our lives also in the lab. First of all, everything has to be scalable to enormous scales. The first hamburger that we made had 1 billion cells, and that was just a quarter pounder. Everything has to be extremely cheap. We eventually want to produce a kilo of material, of tissue engineered material, for roughly $7 or so, or maybe $10, but not more than that. Safety is a little bit easier to balance in food than it is in medical applications. You can imagine that you cannot implant living cancer cells in people with impunity. But you may be able to actually eat dead cancer cells without any problems. That's just an example. Obviously, we're not going to feed people dead cancer cells, but just an example that the safety profiles may be different, and that would hopefully translate into also more economical components that we use to make this. And the last thing that I learned kind of the hard way is that whatever you do with food results in emotions. We are quite emotional about our food, and taking in food is always kind of an issue in terms of safety, long-term safety, and the balance is very different in the food system than in the medical system. So when I started, we basically, and for the 2013 hamburger, we focused on making organoids, a technology that in the medical field by Herman van den Berg at Brown had already been developed for 15 years or so. We just applied that same technology to make a lot of muscle organoids and made the first hamburger. So basically what you do, you take the stem cells, already indicated in muscle and defined in muscle, and all the protocols to get them out were kind of established. So you take the stem cells out, in our case, cows, you let them proliferate, and you put them in a gel and let them form an organoid. The only real difference with the typical organoids that you may be used to is that in this case we need anchor points. So the ends of the muscle, if you like, we do it in a different way, but the ends of the muscle have to be anchored so that when they start to contract, they actually build up tension. Now, in the meantime, this was back in 2013, and in the meantime, quite a few companies popped up, especially in the last 3 or 4 years, and some are kind of along the chain of from a complete muscle to the individual proteins, developing technologies and probably also products. So going down to the individual proteins just by fermentation and making recombinant actin myosin, going up cell-based, which is a term that's now often used, and that kind of suggests that some companies are offering the cells as ingredients in an otherwise plant-based product. Then the fiber-based, such as us, or organoid-based, if you like, such as us. And then finally, there are also now 2 companies or so that focus on the tissue engineering of the full complexity. So not a full thickness muscle yet, but the full complexity with not only muscle and fat cells, but also fibroblasts and endothelial cells and so forth. So it may not be, I'm going quickly through this, may not be as a surprise to you that we are still kind of focusing on those organoids, because we feel that the functionality of muscle protein in terms of taste, in terms of texture, and of course also nutritional value, it basically requires a full maturation of a muscle fiber. So we take a biopsy from a cow, we isolate the cells, we separate them in fractions, and we do that in a more and more sophisticated way to get the pure muscle cells that will stay muscle cells for a long time, but still keep their proliferative capacity and are able to differentiate into muscle tissue. And we separate a subfraction of that same muscle biopsy that we know can actually turn into fat cells quite efficiently. Then we go through the organoid formation stage. So in a muscle, it's a fiber. It first requires fusion of cells and then putting them in a gel and let them form through self-organization, a muscle fiber. And for the fat cells, it's a relatively easy process. You just have to encase them basically in a biomaterial. And then you combine those tissues. We do that separately. We combine the tissues in a hamburger. So the only thing that we do different from Herman van der Burg at Brown when he started this now 30 years ago, I guess, is that we let the cells, when we put them in the gel and we let them form an organoid, we let them do that in the form of a ring shape. And that has a couple of advantages. First of all, once they start contracting and they already have kind of found each other and attached to each other, that translates into tension. And the second advantage is that it's much easier to harvest than when you put them on Velcro or whatever anchor point you're going to use. So that was, even already in 2013, was the way we made muscle cells. At that time, we didn't really make fat tissue yet. So this is kind of a general slide because it's nice to, to be able, it was quite interesting actually in 2013 to present this to the world. Quite interesting in terms of all the reactions that you get to it. But it was still not really a product. It cost us obviously €250,000 to make one single hamburger. And there were still all sorts of components in there, such as serum and collagen and matrigel that you would not want in a food product. So we had to work on quite a few issues to make that proof of principle into something that you potentially can produce. I mentioned the cell selection. We have become more and more sophisticated based on single cell RNA sequencing and crude sequencing, and get more and more profiles of cells in that mononuclear fraction that we get out of our digestion system, and get more and more sophisticated, and we get better and better at identifying the cells that go through prolonged proliferation stages and go into and still be able to differentiate into muscle tissue. So initially we started, this is a project that we started with Qi Jing, a colleague from China, a Ph.D. student, and he translated basically what they had done in pigs to bovine and do a double positive selection for CD29 and CD56, and a double negative selection for CD31 and CD45. And then you see that you have an enriched population of myosatellite cells, which are designated by PAX7 positivity. And if you keep them for a longer period of time, you actually have an improvement over a non-selected cell. And as I mentioned today, we use two other criteria. One is actually one that has been published, is ITGA7, which is alpha-7, one of the other integrants, and the other is PDGFR alpha, the alpha receptor for PDGF. Obviously, when you think about making this a production process, you have to make sure that your input is rather robust. So this is from, I don't know, something like 20 isolations from different cows. And these are the proliferation curves. You can imagine it's quite tedious work, but you have to go through this if you want to establish a kind of a robustness of the process. And you see that those proliferation curves are pretty similar. There's not a lot of variation from animal to animal, or from muscle to muscle for that matter. Mind you, we don't really, most of the samples that we get are from older animals. So it's already kind of a selection and a standardization, if you like. We're currently doing a project where we follow cows over their entire lives, starting from just the day after birth to 2 years, to see whether there's actually a big effect of age and gender and site where we take the biopsy. Now, currently, we are, so this here on the x-axis is the number of doublings that you theoretically could get. I put 50 in there because it's the Hayflick number, which is somewhat a mysterious number, as you probably know. And if you then do the math, if you get through those doublings and you still are able to differentiate those cells, which is a big if, then after around 25, 30 doublings, you can actually get a decent multiplication factor from your biopsy, which is typically 0.5 gram of tissue, to about 2,000 kilos of meat. If you get above, we are currently at that stage. If you get above that, obviously the amount of meat that you can get from a sample exponentially grows. Qijie Ding, who did the CD29 and CD56 sorting, also played around with a P38 inhibitor. This was also already established in the literature. And you do see that through the passages, you see two things. One is that PEC7 positivity actually declines inevitably. This is partly because this was still at the time when our sorting system was a little bit less sophisticated. So this is partly due to overgrowth of the cells that we now wean out in the very beginning, but partly also just loss of stemness, which has been described extensively. And this P38 inhibitor delays that a little bit. Obviously, those inhibitors are not food compatible, so you would have to have safety issues, but it does demonstrate the principle that by tapping into these cell cycle mechanisms and senescence mechanisms, you can actually rescue some of that senescence. I will go, they still differentiate after this P38. It doesn't really affect differentiation, so that's a good thing. We are using, this you all know, so I'm going over this quite quickly. We are using primary cells. There are a couple of reasons for that. One is that we are based in Europe, and in Europe the acceptance of GMO is not really high. So there's already kind of surveys indicating that a combination of cultured meat and GMO is kind of killing for consumer acceptance. So we are staying away from GMO. So we've also stayed away from cell lines and from iPS cells, for instance. But it's a choice that you make. And I'm not really religious about these choices, but mostly for consumer acceptance, we have chosen for the moment to stay away from that. But of course, there are many opportunities to create cell lines, also to create iPS cells without, strictly speaking, GMO or genome integrated transgenics. So there are lots of possibilities out there, but we do feel that with our current system, we can already, reduce the number of biopsies to such an extent that we might not even need this. Of course, you have to scale up cell growth. These cells, as you know, are adherent. They need to grow on a surface. And if you want to transform it from a planar system to a cell suspension system or a bioreactor system that allows you to scale up to large scales, at the moment you're still dependent on microcarriers. So we have played a lot around with all those microcarriers that are out there, and they actually work quite nicely. So it's not really an issue. It's just, in the end, it's cumbersome because you have to separate the microcarriers from the cells, and it's a cost factor. So we eventually want to get rid of this, but for the moment we are banking on the microcarriers that are just available and work in our hands. I don't know whether I have a slide of that, but we can actually grow the cells in aggregates. The fat fraction is a little bit easier to grow in aggregates than the muscle fraction, but they can be grown in aggregates. But we have not really optimized the system to the degree that we can already put this in production. So currently we're still banking on the microcarriers. So if you're not familiar with scaling up, then this is no rocket science. It's you have to go through a lot of motions. Of course, you have to create a cell bank so that you can always have enough supply of good quality cells. And then in order to create the inocula for larger bioreactors, you need to work your way up from a small system to a ever increasing system. So currently we are still at benchtop bioreactors of about 10 to 40 liters, which might, for some of you, might seem a lot. And in terms of surface and amount of cells that you can get out of it is a lot. But in terms of what you need for meat production, it's actually just the very, very beginning. Eventually, the smallest bioreactor that we should use as a unit for production is a 1,000-liter bioreactor, if not higher, if 1,000 can actually cut it. So, this is here are the aggregates. So this is, these are myoblasts, and you see you can make beautiful aggregates of them, and they actually grow. It's still, the proliferation rate is not as high as on microcarriers, and definitely not as high as in planar systems. So we're still optimizing this, and there's lots of room for optimization. If you translate this bioreactor system into what would be the typical meat output of a farm, let's say in the Netherlands, farms in the Netherlands are typically quite small, so 250 cows per farm. So it's kind of low intensity, if you like, and that produces 35 tons per year of meat. And that you can actually recreate with 4 of these 1,000-liter bioreactors. The samples are in the bioreactors. The multiplication steps in the bioreactors take about a week, so you can get a lot of cycles of these things out of in a year. And so that's kind of the equation, if you like. If you translate that to world production, then it would obviously be a tremendous fermentation capacity that you need. Pretty much 1.5 the current present fermentation capacity in the world. So you would have to double it, and then again half of that. So that's quite an undertaking, and obviously we are far away from that, but it also doesn't seem undoable. It's something that is within the realm of, if we can create this fermentation capacity, we can also create 1.5 of that, I would think. Obviously, you know, we're making a food, and it's quite important that this looks like meat, and this tastes like meat, and this has the same bite. I already kind of refer to what my thinking is. This is not proven, by the way, but my thinking is that the functionality of those muscle proteins is very dependent on not only the protein composition, but also the arrangement of the typical myosin and actin proteins in sarcomeres. So in my mind, you cannot get around making the full tissue, but again, some colleagues tend to think differently. These are just a couple of samples of our muscle fibers. These are now completely animal-free, so there's no collagen or matrigel anymore in there. No serum has been used to generate the cells. It's just completely animal-free, and they are as good and as functional as the muscle fibers that we get from, cells that have grown in serum or in collagen, matrigel, or in fibrin. The way we approach the the optimization of all these processes, because that's what it is. If you want to get rid of serum, you have to obviously find a good recipe with growth factors to replace that serum so that the cells keep on growing and also keep on differentiating. And as you probably know, differentiation of muscle cells, of muscle precursor cells, is quite easy. You take away all the proliferation stimuli, and then they kind of start to differentiate, at least in 2D. In 3D, it's a little bit more complex. But obviously, when you do this, you typically in a research environment, you accept that 60% of the cells fuse. We cannot accept that. We have to go for 100% of fusion of the cells, which is quite an ambitious goal. So we are using a lot of omics data to follow that differentiation process. And this is just an example of it, where we have followed kind of that differentiation process, took samples at every different step of the differentiation process, then do, in this case, RNA sequencing, do a lot of bioinformatics on that. And in this case, we identified a couple of receptors that are increasingly expressed when these cells are differentiating. And we had the common, be it somewhat naive idea, that if you then stimulate these receptors with their traditional kind of ligands, you might stimulate the differentiation. So we have tested those hypotheses, and we indeed find that if we add, in this case, for instance, it's IGF and acetylcholine because of the cholinergic receptor, cow growth hormone, and some others also, for instance, T3, the thyroid hormone. So they all help with that differentiation process. Up to the extent here you see the expression of a couple of proteins, the typical muscle proteins, myosin and desmin, and myogenin, and you see that in our serum-free differentiation medium, we are not quite at the level of myosin yet. In 3D, by the way, we are, but in 2D, we are not quite yet. And but it is already improving versus the first kind of iterations of this serum-free medium. So this is the serum-free differentiation medium. For the proliferation medium, obviously we have another recipe. The other animal component that we wanted to get rid of was the gel. As I mentioned a couple of times, traditionally, and for the hamburger in 2013, this was collagen matrigel or fibrin. Those are the traditional gels that you use for that. But we didn't want to do that because, not that we have anything against animal components, but obviously if you want to pursue this, you want to reduce the number of animals, getting substances from the animal that are not replicative is not going to help. So we wanted to get rid of those, and we basically moved to, I will skip this, there are other ways to even improve, and we did this in the past with electrical stimulation, where you can improve the protein expression. I will go over this. But if you, this is myoglobin, which is one of the other kind of functional proteins in there, which colors meat red, and is also a heme iron kind of provision. And it was already known in literature that if you grow the cells under hypoxic conditions, they actually start to express myoglobin. So we grew some of those organoids under hypoxic conditions. Actually, they can sustain quite low oxygen levels, and they start to express this myoglobin. I don't seem to have the slide. Oh, it comes back later. Sorry, the order is a little bit mixed up. I will come back to the gel component later. First, the other component of the hamburger, the fat. We have struggled. Well, in 2013 we didn't have fat, but obviously it was important for taste and for juiciness. So we started to work with different cell lines, mesenchymal stem cells, with adipose tissue-derived stem cells, with dedifferentiated adipocytes, but also with the fibroadipogenic precursor in muscles, the Fab cells. And currently that's our favorite cell for a couple of reasons. First of all, it grows very fast. And second, you can get it from that same biopsy as the muscle. And third, the efficiency of adipogenic differentiation is actually extremely high. So we now have up to 100% of the cells that differentiate into fat cells, and you see here on the right lower corner that they actually become monoglobular white adipocytes. What is somewhat a pleasant surprise is if you do that. This is lipid profiles. This is all the, or at least from 16 to 22 atom lipids, fatty acids. You see below here the profile for two cow samples here in the lower, 2 cow samples, and you see here different populations of cells. And in the population, this is the Fab population, you see that if you get this 100% differentiation, which we routinely get, that fat profile actually starts to resemble that of regular fat tissue from a cow quite nicely. So we're very happy to see those results. We have yet to do the tasting though. How about, this is the section about those components, getting rid of the serum and getting rid of the collagen. So we have, as kind of indicated earlier, through RNA sequencing, but also just trial and error, have developed a couple of recipes for growth of the cells, both the satellite cells and the Fab cells. And we now have actually a serum-free medium that performs typically better than serum itself. We're still working on improving that and getting the price down, which is obviously extremely important. Not immediately related to serum because that's expensive too, but it's really important to get the price down as well. On the gel, so this is what I was saying. We actually have chosen a very unremarkable system, if you like, just an alginate, because it happens to be widely available, it happens to be cheap, it happens to be edible. So, and it's also one of the most commonly used polymers that is used in hydrogels, where, of course, Ali is the world expert on it. And as you know, those have to be functionalized. You cannot really, the cells are happy in there, but they don't really interact with the matrix. They don't attach to it. So you have to functionalize them. And here we also just took the most commonly used functionalization with RGD peptides. And then you do see that the function of the gel that is important to basically allow the cells to grab the polymer structures and rake them in, as a fisherman rakes in its net, to rake them in, squeeze out the water and compact the tissue and form a real water poor tissue. So one of the parameters is contraction or compaction of those tissues, and you see here that with high RGD content, that contraction is actually a lot higher than with the somewhat more traditional lower RGD contents. We have played around with many different peptides other than RGD, many different forms of RGD, different ways of binding it to the alginate structure, and there are quite interesting differences in this particular functionality of alginate. But as I mentioned, currently we are at the state, and I'm extremely happy to say that we get those muscle fibers in a animal-free, with this functionalized alginate animal-free system, as if it were collagen matrigel or fiber. Also here we now have a team of 5 biomaterial scientists, and they look still at different ways to improve that gel. Basically adding secondary components to the gel. In terms of polymers, these can be protein polymers or sugar polymers, or even fat. But every time, of course, we ask the question, is this edible, is it cheap, is it scalable, and is it shapeable, if that is a word. In other words, can you kind of spin it or print it or extrude it or whatever, so that you can actually impose a structure to the gel. We do the same with recombinant functional proteins. I showed a little bit of the laminin. Laminin works beautifully well. It's very expensive at the moment, so it's not something that we envision that we're going to use in a production system, but it does actually work quite well, somewhat to my surprise. We're looking at plant and insect-based analogs of these functional components of the gel and the interaction with pro-differentiation triggers. This is typical tissue engineering, but there are obviously lots of variables. One interesting part, which is kind of the advantage that you have as a company, not only the advantage, also a necessity, to be honest, as a company, that you can develop high throughput systems to do it. So we have increased the speed of our organoid formation from by almost a factor 200 just by automating the pipetting systems and also automating and making the analysis a lot easier. So you saw a couple of confocal and Western blots, but we have developed a couple of ELISAs to actually have a readout system for the structural proteins. One important aspect that you may not think about every day is recycling of the medium. For cost, this is important, but also for resource efficiency. Cell culture is an extremely inefficient system. So here is an example where you have the essential amino acids on the left and the non-essential amino acids on the right. And if you culture cells for 7 days, which is quite long, then you still see, obviously, a lot of amino acids being present in that medium. We throw it away because of ammonium and lactate and so forth, and a drop of pH. Therefore, we throw it away, but the ingredients, the components are actually still there. So it's a very wasteful system to throw that away. So currently we are doing a large, every experiment that we do, whether it's in proliferation or differentiation, is combined with metabolic analysis to eventually derive at one big database that allows us, that informs us when to add amino acids and when to reduce, for instance, ammonium or lactate. And try to minimize the amount of resources that we use for this production system as much as we can. I think it's extremely important, and will also have eventually a lot of ramifications for what is being done in the biomedical field. One of the other things we're doing, and here I apologize for the x-axis where I had to be a little bit secretive about which of those components actually are. What we have done here is use a amino acid free medium. So no amino acids whatsoever, and we basically have sourced a large number of hydrolysates from pea, rice, cotton, wheat, soy, all the sort of the hydrolysates that you can find out there that are provided by large feed or food suppliers, if you like. And basically replaced the amino acid component of the medium with these hydrolysates. And here, the 100% line is actually equivalence with the amino acid, with the traditional amino acid composition of, in our case, DMAM-F12. And you see that some of those hydrolysates, for instance, in this pH6 fraction, they actually are as functional as the traditional amino acid composition, which is interesting in a way because these hydrolysates they're quite crude. We don't know exactly what the chain length is and what the distribution of amino acids is and so forth. So there will be some single amino acids present, but there will also be dipeptides and tripeptides and oligopeptides. And we think that we actually don't need to feed these cells single amino acids. We can actually feed them di or tripeptides or longer chains that they can use with their specific enzymes for incorporation into muscle. So this is a large-scale endeavor with a lot of, interestingly, a lot of feed suppliers, animal feed suppliers that produce these hydrolysates and that are also in our sort of collaborative sphere where we screen all these hydrolysates. This is important for the price of the medium because the medium, culture medium, is 80% of the price of the cost of cultured meat. 70 to 80%, which by the way is the same for animals. The feed is also 70 to 80% of the price of meat, regular meat. And if you break that down into consumables, then and in particular the medium, of course, then in the case of FBS, it's expensive because FBS is expensive. Well, we have many reasons to get rid of that, so that we have done. Then the next factor that becomes expensive is growth factors. That's a very interesting field. I don't know who of you had read the report of GFI or who actually believed that report. But currently, as you may know, we are paying about €1 million per gram of FGF2. FGF2 is a very simple protein that is recombinantly made and not dissimilar from enzymes that are being made in the feed industry through recombinant technology, for about €10 per gram. So we are paying €1 million per gram, and we never buy a gram, of course, because we use so little of it. But actually the price is, if you think about it, is quite ridiculous. And it has to do with, that it's pharma grade and highly pure and all those type of things. But if you really think that whether you need that for food production, then that's probably not the case. So there are currently about 15 startup companies that are trying to generate recombinant proteins at the level of somewhere around €10 per gram. And then because the low quantities that you use, it's no longer a cost factor in the medium. And then of course, the next cost factor will be albumin and transferrin, similar kind of, it's a little bit more difficult to make, but similar considerations. And then you go down to the amino acids. And that's where those hydrolysates come in. So if you don't do the hydrolysates, just do a reduction of growth factors, prices, no serum, scaling up, and recycling, you end up with a price of €150 per kilo. So roughly $160 or so, $170 per kilo. That is still expensive as meat. For a tissue engineered product of a kilo, it's actually very, very cheap. But to get from there to that price of regular meat, you would have to go to individual components like the sugar, the amino acids, and all the ones that will then become the cost limiting factor in the medium. So as I mentioned, we are looking currently at a lot of these hydrolysates from peas, from wheat, from and also the sugar component from basically starch or other sugar components. One very interesting, by the way, recent result that we got is that by changing the sugars, we can actually pretty much avoid a large part of the lactate formation. So, which is kind of to be expected, but you can do that without having an impact on cell proliferation and cell differentiation. All right, then the emotional part, I will go over this very quickly because it may not be too much of interest to you. Yes, you have to deal with it. There's a lot of surveys that are out there already, and we have done some investigations ourselves. We are biologically programmed not to eat stuff that we don't know. That's the bottom line of it. And it's a very functional kind of mechanism, but if you want to introduce a new food, it's not that helpful. So this is an interesting one. This is actually off Niagara Falls. So there are a couple of studies. I will not go into this study because time, and I want to reserve more time for questions. But we have done a number of studies ourselves where we actually have people eaten this and reflect on it and see whether the amount of information or the type of information that we give affects their acceptance. And interestingly, giving information does drive acceptance, but what that information is doesn't really matter that much, which is somewhat surprising. And by the way, everybody in this particular experiment who got presented with a piece of hamburger that was labeled as cultured, they all ate it. You know, again, it's in a setting of a study in a university, so it's not really, maybe not reflecting that much what people are doing in a supermarket, but it is at least telling to some degree. Okay, I will go over this. This is a little bit of an overview of surveys that have been done since, well, actually 2011. And here the blue bars are actually the people who say, well, yes, I accept this. This makes sense, and I would start eating them. And you see that this is roughly around somewhere between 25 and 50%. And you also see over time, if you if you would draw a line through this, you would see a gradual increase in acceptance. And you also see that it's not really that dependent on geography. So those surveys have covered the US, the UK, EU, Australia, India, China, and the numbers are somewhat the same. So in surveys, the outlook is kind of good. This is a novel food. Novel food is actually an expression that the EU uses for something that is not sufficiently equivalent to food that have been eaten before 1997, when this law came into effect. And that means that you have to provide evidence that this is safe. In our case, in Europe, it's EFSA. In US, FDA and USDA struggled quite a long time. Who is going to provide the oversight? And they last year they came to an agreement that they're going to do that jointly, which everybody from the outside could already have predicted, but it took them some time to do that. So they're now also starting to describe what would be required to get it approved by the FDA, USDA. In December, the first product in Singapore has already been approved. So in Singapore, you would now be able to get chicken samples made by Just from the US. We started this very early, and I'm very happy to say that currently there are about 40 to 50 companies, startup companies working in this field, quite a few actually in California, but pretty much spread out over the world. Israel has a high density. There are a couple in Europe, a couple in China and India and Australia. So it's scattered, but it's around 50 people, for 50 companies. Still, all of them startups. There are no products really on the market other than the samples that are just recently came to the market in Singapore. But a lot of money is actually flowing in this, and right now our company has 70 people working on this, mostly scientists like yourselves in quite similar settings. We are probably the second largest company, but currently we already have worldwide, I'm estimating, so 500 scientists or so working on this endeavor. So, you know, I hope to have convinced you that this is worth pursuing, that this is important to do. I didn't give all the reasons why we're doing this, but it's quite obvious. It's food security, environment, and animal welfare. And if you have followed the newspapers, you could have, you can have this information. Fat production is an important part of the whole concept. We didn't have this in 2013, but now we do. And actually, that's looking very good. Scale up, obviously, is a formidable task, as is reducing the cost to a price that consumers are willing to pay. And regulatory approval is required. Personally, and I really speak for myself, I think this is a really fun application of tissue engineering with a lot of potential kind of interaction with the medical field. And I would encourage you all to seek that interaction and to try and learn from each other. The challenges are somewhat different, but the techniques are actually quite similar. So this is already an older version of our crew. As I mentioned, we have grown very, very quickly. And so we're now working on this with 70 people who have provided some of this data. So with that, I would like to end, and I'm happy to entertain questions.
Thank you for the excellent talk, sir. One question is, what would be the cost versus scale kilogram per day? What scale size would bring cost comparable to meat?
Well, as I mentioned, those bioreactor scales of at least 1,000 liters would be absolutely necessary. Below that, it's almost undoable to get it to become cost effective. And so production scales, I think production scales like that regular farm of 4,000 liters in total could actually become price competitive. Above that, there is still economy of scale, no question, because you reduce labor and things like that. But that's really incremental. That goes, you know, from €5 per kilo to €4.80 per kilo or something. So that for a commodity will be an important step. For us, it's kind of an incremental step.
I see. What differentiation media do you use for fibroblast? Regular one used for adipogenics of stem cell, can we know?
It's based on MSC6.
Okay.
But we do add a lot of, I imagine there are two parts of this question. One is the medium, and that this is based on MSC6, but we have modified it, and we're currently applying for a patent for that recipe. The other is the cocktail required to differentiate adipocytes. Traditionally, this cocktail includes IBMX, dexamethasone, insulin, and in some cases rosiglitazone. those compounds you cannot use. You cannot use for a food application. That's almost impossible. Definitely not rosiglitazone, and definitely not IBMX because they're toxic. So we have replaced that with other triggers, and basically triggers for PPAR gamma and CPEB.
Okay. What is the estimate cost of your serum-free media? Is it kind of commercially available, or did you formulate it?
No, it's not. Well, the base is commercially available, of course, because it's just DMEM or DMEM-F12. But the cocktail that we're adding to it is not commercially available. That's kind of custom made. The cost currently is, I believe, about roughly €100 per liter.
Okay. What is the cost of the alginate RGD, and how does it taste?
Oh, the cost, quite frankly, I don't know. It's a good question. We haven't got really to that cost factor yet, so I don't know. We make it ourselves. We don't buy it. We make it ourselves. Alginate is very cheap. You have to be a little bit specific about the molecular weight of the alginate, but it's cheap. The RGD, of course, is also not expensive, but the entire process of doing it yourself and spending time on it and all that sort of thing eventually ends up with a cost factor. But quite frankly, I don't know. We may have it, but I don't know it by heart. The taste is actually insignificant in this case because on a weight basis, it's such a small, tiny component of the entire product.
Okay. What are the checkpoints to assume that we are in the right direction? Namely, should we check protein expression test? How can we merge the quality of the product at each step? Finally, in the final product, does the ratio of biomaterials to myocells matter?
Yeah, to start with the last question, yes, it matters, of course. And this is all the information that we are providing to EFSA. What is the compositional analysis, and then the alginate is part of that, and the fatty acids are part of that, and the muscle proteins are part of that. So yes, both for the fat and for the muscle, the contribution of alginate to the final product is, as I mentioned, marginal. What you will have to do is, because when you ask about control points or checkpoints, that's something that in a process and in a food production process, you have to have these checkpoints. You have to have quality assurance. So one of the quality assurances is genetic stability of the cells, which is kind of obvious. And the other is indeed quality of protein, both the composition and the amount. So the amount, none of these, by the way, are currently specifically defined by the regulatory bodies. So we kind of have to start with our own definitions and then hope that the regulatory bodies actually will agree with that. In terms of protein amount, and also to some degree protein composition, this is not so much relevant for safety, but it's relevant for being able to label it as meat.
Okay. What are the major microbial safety issues for cultured meat?
None that I can think of. The only real threat is if people, especially in these large-scale cultures, start to use antibiotics. The tendency in the entire industry, and definitely in our case, is to culture the cells in the absence of antibiotics. And actually that works fine. The cells grow better in the absence of antibiotics, and we really don't see any increase in infections. So you have to, so that's important. Obviously the starting material, it's a biopsy, it's taken under sterile conditions from a cow as much as you can do that. And so we don't really have infections, to be honest. What you will need to do, and this is a special kind of safety aspect, is you will need to make sure that your donor animals are disease-free.
Okay. How close are you in texture and taste aspects? What are the challenges in these areas?
Right. In terms of taste and texture for the muscle, we are fine. This just tastes like muscle. And by the way, every hamburger that you have, unless you have made it yourself, but you buy, actually has flavoring added to it. So we are working with a flavor company to do that gap analysis and to add flavors to if there is any gap. For the fat, we yet have to do the tasting. We just send samples to the flavor company to taste them. This has to be professionally done. Also here, there will be a gap analysis, and the flavor company will actually add flavors if necessary. The biggest concern in the industry is not the absence of flavors, but the presence of flavors that are unwanted. That has been in the plant-based industry also the biggest concern, and actually you need to do quite a bit of work to mask those off notes, those off flavors, if you like. So far, we have not experienced them in these cell cultured products, but it's still something that you have to do.
What are the manufacturing challenges to achieve texture in more complex cuts of meat, such as ribeye? How far away do you think we are from such products?
I think far away, to be honest, and probably you guys know this as well as I do. In order to make that happen, so the way I'm thinking about this, we don't need to make a tissue that's thicker than, let's say, an inch or 3/4 of an inch, but still that's a very thick tissue in most of our minds. So this requires three things. One is obviously another biomaterial that is stiff enough so that it can direct all those fibers in a specific plane, because it's a very regular tissue. So biomaterial, in addition to the gel, biomaterial that's stiff enough to direct all those cells in the same way. Second is that you have to co-culture at the very least the muscle cells and the fat cells. Currently we're doing that separately, and you have to have some fibrous tissue in there as well, of course. And third, as we all know, you would have to have a channel and perfusion system to keep everything alive. In my mind, I'm a vascular biologist. I worked 30 years of my life on blood vessels, but I don't think we need specifically blood vessels for this. I don't think they add too much to the taste or the of the meat. But you do need a channel and perfusion system for oxygen distribution, nutrient distribution, and waste removal. What I think the biggest challenge will be, and nobody actually has ever pointed to that, is if you start creating a tissue, and the way we have been developing these tissues, they all compact. If you do this in a large 3D structure, compaction leads to, sorry about this, compaction leads to holes in the meat, right? Like Emmentaler meat. So in my mind, you would have to have a system where you can either prevent making these holes or fill the holes later on with additional cells and additional material.
What do you suggest for interdisciplinary teams of tissue engineers and food engineers to focus more on?
Oh, that's a very general, very general question, I guess. You know, for what is important for us is that you steer away from the very complex proteins, because they are, even if you make them recombinant, they are, there will be expenses associated with them. So I believe strongly in providing the viscoelastic properties of your materials, where the cells are happy with those materials and provide as short chain kind of peptides that mimic the different functions of the larger peptides. So all of these peptides, the laminins, the fibrins, the collagens, have all a kind of different functions associated with different smaller fragments of those peptides. And in most cases, you can actually define a short fragment that mimics that function.
Okay. Could you explain us the regulatory process to get certificate to bring it to the market? And what are the main challenges to get FDA approval for this innovative food product?
The biggest challenge is that nobody has done it yet, and the FDA doesn't really know how to do this. All these applications are based on single components, on food additives. So basically doing a toxicity test and an allergenicity test, but this is a complete tissue, so they don't have really processes for this. The way it's approached is that you are quite reasonable thinking, basically, that you do a compositional analysis that for sure you know that there are no components in there that are toxic, or if they're in there, there are such low levels that they don't issue any threat. You have to make sure that your process is robust enough. That's true for every food production, so that from batch to batch you can actually have a consistent composition and consistent quality. And then there are regular things like allergenicity tests and so forth. You have to be quite specific in how the food is going to be used. So you want to provide that information on the raw material, on cooked material, on barbecued material in combination with other foods that you all have to identify. And then what you don't have to do, at least so as far as we know, you don't have to do animal testing or clinical trials, which makes me very happy, because they would be completely nonsensical to do, and probably not even, you probably cannot even execute them. But that's most likely not required. So you would have to do some what we call ADME, so digestion and absorption and metabolysis. But if the components are already known in terms of their ADME, you don't have to specifically do those tests in animals or in humans.
Okay. When do you foresee these products hitting supermarket shelves? And do you think this technology will completely replace the meat poultry industry? If yes, by when?
Right. So first question, when do they hit the shelves? For that, you have to have a reasonable price, right? So inevitably, these products, like you see in Singapore in the beginning, are going to be exclusive because there's not enough production, and they're going to be expensive because we haven't really gone through all those steps yet of reducing price. So what you will see now, particularly in Singapore this year, a couple of other products will become approved from different companies. And then I think the heat is on, and in the US, a couple of applications, and in the EU, a couple of applications will be submitted. In the EU, the process takes 1.5 years. So for that, in the EU, the citizens have to wait quite a long time for it to be approved. In the US, I imagine it can go quicker, but we don't know that yet. So my guess is in EU and US, we probably see in the next 2 years, we see at relatively small scale, relatively high price products in restaurants. For supermarkets, I think it will take a couple more years for the production to be scaled to that degree, and for the price to come down so that so it's actually a reasonable choice for people. The question, if this is going to replace meat, traditional meat production, it's interesting because my PR department doesn't want me to say this, but you know, that's I don't always listen to them. I think it actually will. It has the potential to transform the meat industry completely and eradicate livestock meat production. And the reason why I'm saying this is mostly based on our shifting ethics towards animal. So currently we eat meat because we love it, and we accept that we kill animals for that, and we know, all of us know that, you know, this is somewhat of a problematic system, but we stash that away in a dark corner of our conscience because we love meat so much. But once you have a credible alternative that is of sufficient quality and of low enough price, we can no longer do that. You see in animal experimentation, for instance, and people who have been long enough in this field like me to have watched this over time, that ethical position towards animal experiments is changing and has changed over the last 30 years and keeps on changing. And eventually this will also happen in the food world, where, you know, one of the, this may be too long an answer, but one of the examples is we had to, in order to do biopsies in animals under anesthesia and sterile conditions, to do biopsies in cows, we had to define an animal experiment. So we are subject to the very stringent laws and registrations of animal experiments, where nobody who cuts the horns of animals has to do that. So we are measuring with different measures for agriculture and for animal experiment, and eventually that can no longer hold. So my feeling is that especially those ethical shifts eventually will eradicate conventional meat production. But again, that's my personal view. It's not science, it's just, you know, try to think this through.
Well, that's all the questions I have. I don't want to take more of your time, but it's excellent talk. Thank you so much for joining us, Dr. Post.
Well, welcome, and thanks for having me and being, you know, attentive at this hour.
Thank you very much, Mark. Really appreciate it.
Welcome, Ali, and we'll keep in touch.
Sounds great. Have a good one.
Okay, take care. Bye-bye.